Welcome students to section 3 of this module. Myself Dr. Divya Bhagavan. Today I will take you through the topic cholinergic neurotransmission. So cholinergic neurotransmission is where acetylcholine is used as a neurotransmitter for transmitting impulses. So this occurs in autonomic preganglionic nerve endings, postganglionic parasympathetic nerve endings, some of the sympathetic postganglionic nerve endings, and somatic motor nerve ending, and nerve endings supplying the adrenal medulla. So cholinergic neurotransmission can be explained in basically 6 steps. Okay, so here you have the cholinergic nerve terminal, presynaptic cholinergic nerve terminal. This is the axon. Here you have the synapse, and here you have the effector organ, which you have, where you have the postsynaptic receptor. Now, this cholinergic nerve terminal. Okay, so coming to the steps, first step is synthesis of acetylcholine. Okay, so acetylcholine, it is synthesized using choline and acetyl coenzyme A with the help of the enzyme choline acetyl transferase. So acetylcholine is synthesized by the acetylation of choline, and this step is catalyzed by the enzyme choline acetyl transferase. So here you have the cholinergic nerve ending. Choline from the extracellular fluid, it is transported into the axon of the cholinergic nerve terminal using a transporter. So this is a rate limiting step, and this choline, it is acetylated with acetyl coenzyme A with the help of the enzyme choline acetyl transferase with the leading to the synthesis of acetylcholine. Once it is formed, it is stored in the storage vesicles. For that, it is taken up into the storage vesicles using another transporter, which is called as vesicle associated transporter. And it is taken up into the storage vesicles and it is stored here till when let's say this third step, where there is release of neurotransmitter. When an action potential reaches this or arrives at this cholinergic nerve terminal, what happens? There will be influx of calcium. Okay, so there will be calcium channels opening. There will be influx of calcium. Once there is influx of calcium, this storage vesicles, the membrane becomes unstable. They will go and fuse with the terminal membrane of the cholinergic nerve ending, and there will be release of the neurotransmitter, that is acetylcholine, into the synaptic cleft. This is the synaptic cleft. So acetylcholine is released into the synaptic cleft, and once it is released, it can go and bind to the receptors. There are postsynaptic receptors on the effector organs. Okay, so you have cholinergic receptors like muscarinic receptors and nicotinic receptors. We'll be dealing about the receptors in the next video. For now, you have to understand that there are postsynaptic receptors on which acetylcholine will go and bind and will produce a response. Next step is degradation of acetylcholine or termination of action of acetylcholine. So this acetylcholine is rapidly degraded or rapidly metabolized by the enzyme choline esterase or acetylcholine esterase, it is called, to produce choline and acetate. So choline is degraded by the enzyme acetylcholine esterase to produce choline and acetate, and this choline can be recycled, okay, can be utilized for the biosynthesis of more acetylcholine. So these are the steps in cholinergic neurotransmission. One is synthesis of acetylcholine. Second is uptake into storage vesicles, where it is stored in the storage vesicles till there is a action potential arriving at the cholinergic nerve ending, and there is release of neurotransmitters into the synaptic cleft. After it is released, it goes and binds to the receptors and produces an action. Next step is degradation. They are rapidly metabolized. Acetylcholine is rapidly metabolized by acetylcholine esterase enzyme to produce choline and acetate, where choline is recycled. This is again the another figure where you can see the choline transporter here. This is the rate limiting step where choline is taken up into the cytoplasm. This transporter is inhibited by group of drugs called as hemicholineums. Okay. So when hemicholinum can bind or inhibit this transporter, there will be a gradual fall in cholinergic transmission. Another transporter I discussed was called as VAT, where acetylcholine is taken up into the storage vesicles and it is stored along with ATP. So this is called as VAT, vesicle associated transporter. This transporter is inhibited by the drug which is called as vasamicol. Okay, so with vasamicol inhibiting this transporter, there will be neuromuscular block which will be developing slowly. Now, third drug is botulinum toxin, which will inhibit the or prevent the release of acetylcholine from the cholinergic fibers. So, the three drugs which can inhibit different steps of cholinergic neurotransmission. Now, coming to the termination of action, I discussed acetylcholine is rapidly metabolized by the enzyme acetylcholine esterase, or abbreviation is ACHE. Okay, so acetylcholine esterase are of two types. We have true cholinesterase, which is mainly present in the neurons and the neuromuscular junction, where you saw the synaptic cleft, right? It is mainly present in the synaptic cleft of the neuromuscular junction, where it can degrade or metabolize acetylcholine and other acetyl esters like methacholine, but not succinylcholine. Whereas the another type you have is pseudocholinesterase or butyryl cholinesterase. It is also called as plasma cholinesterase. It is present in the plasma as well as in the liver. It is mainly responsible for the degradation of succinylcholine. It can also degrade acetylcholine but very slowly, but not methacholine or other acetyl esters. So with that, we come to the end of cholinergic neurotransmission. We studied about the six steps where acetylcholine is synthesized, it is stored, it is released, it can bind to the receptor producing action, and it is degraded by acetylcholine esterase enzyme, and the choline can also be recycled. Thank you for actively listening.